Welcome back to another session of RNA biology. So we were here in the previous class, we were talking about the 5' cap. So 5' cap is a structure that stabilizes the RNA from degradation. So it can be any molecule means not any random molecule. There are different possibilities of 5' capping exist, but in general it is a 7-methyl guanosine cap that exists as the 5' cap in every RNA molecule. So eukaryotes have around 5 different types of RNA polymerase: polymerase 1, polymerase 2, polymerase 3, polymerase 4 and polymerase 5. So RNA polymerase 1 is predominantly in the nucleolus. Nucleolus is a structure that can be seen densely stained in the cross section of a cell, etc. You can never see a nucleolus lighter in stain. It is because the ribosomal RNA production is quite high in the nucleolus, because of which there will always be a intense staining. Now RNA polymerase 2, which is mainly in the nucleus, throughout the nucleus, and the nuclear pre-mRNAs are transcribed with the help of RNA polymerase 2. RNA polymerase 3 is mainly transcribing transfer RNA and the 5S ribosomal RNA. RNA polymerase 2 is mRNA, RNA polymerase 1 ribosomal RNA with an exception of 5S ribosomal RNA, and RNA polymerase 3 is handling tRNAs, 5S ribosomal RNA and other small nuclear RNAs. Whereas polymerase 4 and polymerase 5 are a feature in plants, and what they do, they produce siRNA. They are mainly meant for silencing some of the mRNA transcripts. In plants, the gene regulation through RNA mediated gene silencing is quite high. That allows protection from invading pathogenic bacteria and viruses. So polymerase 5 is also undertaking siRNAs and some non-coding RNAs which can help in silencing of the target genes. And if you look into a typical RNA polymerase 2 is well studied as a candidate gene, simply because the gene regulations are more applied to RNA polymerase 2 than polymerase 1 or polymerase 3. It is because a cell have got its identity mainly because of the total number of protein, total diversity of protein present in that cell. So RNA polymerase 2 being the polymerase considering or doing the transcription of gene encoding mRNAs, its transcription or its study or its regulation becomes more important than polymerase 1 and polymerase 3. So in a cell there are group of proteins called specific transcription factor or tissue specific transcription factors that allow where a polymerase 2 should bind and start its transcription. If you look into a typical RNA polymerase 2 promoter, you can see a +1 sequence, which is the place where the transcription initiation happens. That is the first base of the RNA, or this is a picture of the DNA. So the first base of the RNA encoded on the template strand. So that is what +1, and downstream of that you have +2, +3, +4 like that. Upstream of that you have got -1, -2, -3 like that. Up around -20 region you have the Tata box, and Tata box have a consensus sequence TATAAAA. Consensus basically mean something predominantly seen or commonly seen. That is the meaning of consensus. This doesn't mean that every gene must have only this sequence. It will allow maybe one base alteration, etc. But usually Unlike the neighboring parts, this consensus sequence are more or less going to be the same. And much upstream, up around -40 region, you have a GC box which has a sequence of GGG, CGG. That is the consensus. And much upstream of that, you have CAT box, and it has a consensus of GG, CC, AA, TCT. And much upstream of that, you have another GC box that is having a same sequence as the GC box you saw earlier, that is GGG, CGG. That is the consensus sequence of a GC box. And then much upstream of that, you have an octamer box. An octamer box have a sequence of ATTT, GC, AT. That is the consensus sequence. So this comprise the so-called promoter region or promoter sequence. Why this is important? Because around 200 base pair long region can contain the necessary elements required for acting as a promoter, and it can stretch up to around 10 kilobases. In some rare cases, the promoter region can stretch up to megabases. That means 1000 kb. 1000 base, 1000 nucleotide is 1 kb. 1000 kb is 1 MB. So it can be stretching much upstream also. So we cannot define a specific boundary common to all the genes. It varies from gene to gene. So if you see initiation of transcription by RNA polymerase II, it starts with one component of the RNA polymerase. There are 12 different subunits are there in the RNA polymerase complex. So TF2D is a initiator molecule of the RNA polymerase II transcription. It binds onto the Tata box region of the promoter. And transcription factors are nothing but proteins that help or modulate the transcription of mRNA in this case. And they are called basal transcription factors. Basal transcription factors are essential. They are inevitable for the transcription to take place, and they usually bind pretty close to the transcription initiation point. And there are many other factors that contribute to the regulation of transcription. They are called enhancers and silencers. Enhancers accelerate the transcribability of a gene, and silencers reduce the transcribability of a gene. TF2D is the first factor that binds, and that is further assisted by other factors. And TF2D is also called as TBP or Tata box binding protein. This is joined by TF2A, and this is further joined by TF2B. So each of these factors are joining sequentially. All these factors cannot join together because that will cause a lot of steric hindrance and disturbance, and also each of these factors need to have a specific stoichiometry in the DNA, which is possible only when one factor is bound. If TF2A is coming to a promoter at first, then it may not find this spot attractive or find this spot worth to start a transcription initiation. So the presence of TF2D is very important for the TF2A to find this place attractive. And then TF2F together with the RNA polymerase II causes the enzymatic activity, that is the DNA unwinding activity. Then TF2E joins that binds to downstream regions of the transcription initiation region. Then to TF2H, which has got helicase activity, and then TF2J that has got a binding capability to the downstream regions. Then comes helicase activity. So helicase activity is an important feature that is required for the strand separation of two DNA strands, without which you cannot access one template strand which makes a copy of the DNA. DNA RNA copy from a DNA template. So RNA polymerase 2 has to have the unwinding activity, and then upstream of that it has to continue with the rewinding of the opened up DNA double helix. So RNA polymerase bends and wraps the DNA, and TF2F alters the non-specific DNA binding by RNA polymerase 2. So it also imparts some specificity to the region, and this results in substantial DNA unwinding but not DNA strand separation. Strand separation, it can be facilitated if there is an unwinding happens. So like we already know, DNA is not like a railway track. Although it is double stranded, it's not like a railway track. It is bent around like a spiral staircase. So you have to unspiral it so that it will look like a railway track, and now the strands has to be separated. So that is what happens as a step 4 TF2F joins, and that unwinding activity is very important for the polymerase 2 to initiate, and then TF2E binds to the pre-initiation complex. And the RNA polymerase 2 you can see as a big yellow color structure here you can see, and the other factors are small factors like A, F, E, B, etc. and D. They are all present in this complex. And then comes the most important step, that is the CTD phosphorylated initiation. How does this happen? We will see them in detail. TF2H has got the helicase activity, we already know, and it also have got a kinase activity. What is kinase activity? Kinase activity allows the protein or an enzyme, what you are talking about, can catch hold of a ATP molecule and add the phosphate group to a target. And in this way, what happens? The molecule will get energized and it can change its conformation, and it can behave in slightly differently than what it is already doing. So the kinase activity is very important. So when the RNA polymerase 2 binds to the complex, it initiates the transcription, which is the complex made of TF2, D, A, H, like that. So the phosphorylation of the C-terminal domain, that is called CTD, is required for the elongation to begin. So what is C-terminal domain? C-terminal domain is the carboxy terminal domain. Every protein have got two domains. One is an N-terminus and another is a C-terminus, because every amino acid have got an amino group and every amino acid have got a carboxyl group, just like the 5' and 3' end. So every protein starts with a NH2 end, that is where corresponding to the 5' end of an RNA, you have got a NH2 that is located in the N-terminus, and towards the 3' end or towards the end of the protein, you have got a carboxyl group. So that is called C-terminal of a protein. So we call it as CTD, carboxy terminal domain. So all eukaryotic RNA polymerase have about 12 subunits, and they are aggregates of the order of 500 kilodalton, and this is a pretty large molecule, and some subunits are common to all three RNA polymerase, like polymerase 1, polymerase 2 and polymerase 3 can have common subunit, and the largest subunit in RNA polymerase 2 has a C-terminal domain, and we were talking about the kinase action onto the C-terminal domain for the initiation or the movement of the transcription. So the carboxy terminal domain, it consists of several repeat elements of a heptamer. Hepta means 7. So you may have heard about this mono, di, tri, tetra, penta, hexa and hepta. So this heptamer is consisting of a unique set of amino acid that is present in the C-terminal domain. So what you can see, that the C-terminal domain, which consists of multiple repeats of 7 amino acid, they are unique. And what are they? Tyrosine, serine, threonine, these are repeated up to 7 times, and these ones are which is going to be important for the phosphorylation event. So this is an RNA polymerase, that is multiple subunits are seen, and the CTD is specifically seen here in the largest molecule in this. This is a Western blotting YSPTSPS. So these are single letter code for different amino acid like tyrosine Y, S for serine, and P for proline, and T for threonine, S for serine, like that. It continues and that repeats multiple times. In yeast it is around 26 times, in mouse it is around 52 times, like that. So this domain are very important for the phosphorylation event, without which the RNA polymerase will not go downwards or downstream. So now if you look into the RNA polymerase 1, we have seen RNA polymerase 2, we can look, is there any unique feature in the RNA polymerase 1? This is called a bipartite promoter. That means it has got sequence elements upstream as well as downstream, and this promoter has a core promoter region and an upstream control element, UPE. So upstream control element and the core promoter both are important for the functionality of the polymerase 1, but it is less complex compared to polymerase 2. And this transcribes, RNA polymerase 1 transcribes ribosomal RNA genes except 5S ribosomal RNA gene, and this core promoter consists of -45 till +20. So it is interesting to note that even after +1 the promoter region is expanding in this case of polymerase 1, and it has got GC rich and AT rich regions, and they also have got specific inter-initiator regions, and that allow the specificity to these promoters. And the binding factors are typical as you saw in case of RNA polymerase 2, and this complex are formed by transcription factors, especially the TBP. That means data binding proteins becomes handy even in polymerase 1, and its cartoon is shown here in the left hand side. You have this DNA and there is a start point here, and it has got an AT rich immediate upstream region, because the reason because that place is much easy for opening up, just like you saw in the case of transcription termination. AT rich region in the promoter allows the opening of the DNA strand much easily, or the separation of DNA strand much easily. And immediate upstream you have a GC rich region, and reason is quite obvious, because GC rich region do not allow further opening of the bubble. You don't want to fall apart, so you want to have some some strength. So AT rich region, upstream of that you have a GC rich region, so that it do not get opened up further. And then you have got the upstream promoter element which can spread much upstream, which is called basically an upstream control element. And once it is bound, these protein factors are bound, then it can form a loop. It can form like a knot like structure, which in turn attracts more and more transcription factors, and then it can act like a holoenzyme, and this core binding factor facilitate the transcription initiation in the case of polymerase 1. And now if you look into polymerase 3, it uses both downstream and upstream promoter sequences, and polymerase 3 has 3 different types of promoters are there, like you can see type 1, type 2 and type 3. It is rather simple compared to polymerase 2 promoter. And RNA polymerase 3 transcribes predominantly transfer RNA and also 5S ribosomal RNA, and this core promoter are seen in different names called boxes. Box A and box C is seen in type 1, box A and box B is seen in type 2, and there is specific domains are seen in the case of type 3, that is an oct domain and PSC domain and the typical Tata box. They are seen in type 3. So transcription factors are quite unique in the case of pol 3, because they can identify different boxes based on which the subcategory of pole 3 promoters are present. So if you look into RNA chain elongation, RNA chain, so far we have seen chain initiation, that is the assembly of transcription factors are important for the chain initiation, and the elongation, when it comes about the elongation, this require the phosphorylation of the CTD, without which it will not kickstart. So the existing DNA polymerase upstream has to retain its double helical structure, and then downstream it has to again retain its double helical structure. In the middle it opens up, the transcription bubble happens, and you can see the RNA polymerase have got some lumen, a loop region through which the RNA, growing RNA transcripts comes out, that you can see in this green color. The DNA strand is shown in blue color, and there is a specific opening through which the ribonucleoside triphosphates enter, that is the raw material for the polymerization event. And there is also a groove for the entry and exit of the DNA. So it has got grooves for opening of the DNA entry and exit, and also opening for the RNA exit, and also entry for the polymerase polymerase raw material, that is the ribonucleoside triphosphate, that is the raw material for the polymerization event. And the important thing we need to know, that is the soon after the production of the RNA hasn't completed. Soon after the RNA polymerase is released from the complex, it need to have the 7-methyl cap. So early stage in the transcription of a gene, in this case it is RNA polymerase 2, it has to undergo capping, without which the newly formed RNA will be degraded. So you can see in this picture, this yellow colored bubble is the transcription bubble and the RNA polymerase 2. You can see the DNA opened up, and then the nascent RNA that has come out, and this RNA has to undergo the capping event. And how does it happen? So at the final step is the cap. So to start with, it starts from step number 1, and you have a utilization of GTP, and you have to use guanylyl transferase that will convert GPPP NP a intermediate, which is acted upon by guanine 7-methyl transferase, and you end up getting a methylated GPP NP. What is this GPP indicates? That means it has a guanosine with all the 3 phosphate intact, and downstream you have a nucleotide, phosphodiester backbone, again nucleotide, phosphodiester backbone. That is why NP stands for. Then a third enzyme acts, that is 2'-O-methyl transferase, which allows another methylation onto the first base, that is written as N. N stands for nucleotide, first base. So there is a methyl group, and this already had a methyl group, and this is now added as a cap, which we call it as 7-methyl guanosine cap onto the RNA. So it is a multi-step process. 3 enzymes are involved for the addition of a 7-methyl cap onto the RNA. So then comes RNA chain termination. So the termination signal has to be very specific, because the specific DNA sequence can span around 1000 to 2000 nucleotides, and there are specific sequence that can act as a termination signal. That is, what are they? AAUAAA, that is one termination signal, or ATTAAA. So TT can be UU in the case of RNA, or when it is in DNA it is T, when it is in RNA it is U. So these sequences have to be present, which will be recognized by the endonuclease so that it can chop at the specific location, so that the growing RNA can fall apart from the transcription bubble. And poly-A polymerase is next enzyme that comes handy for the 3' elongation, 3' end elongation. So pole 2, pole 1 and pole 3, all of them have to be having a proper termination event. without which the transcription will continue. Let us see in detail. So 3' poly A tail has to be formed soon after the transcription termination. So there is a 5' 7-methyl guanosine cap is there, then there is a AAU AAA signal is there, and soon after that, around 16 to 18 base downstream of this signal, you have an endonucleolytic cleavage happen, and much downstream you have a GU rich sequence. GU rich sequence will prevent further movement of this transcription bubble, and as soon as the RNA is cleaved, then it has to be recognized by the poly A polymerase, so that downstream of this poly A signal, AAUAAA, that is poly A signal, that is recognized by the poly A polymerase, and it will add a poly A tail. Remember to distinguish between poly A signal and poly A tail. Poly A signal is a sequence that is unique, as you saw here, AAUAAA or AUUAAA, or in DNA it is AATAAA or ATTAAA. So only two sequences are predominant poly A signal, which is recognized by the endonucleases to cleave, and which in turn is recognized by poly A polymerase for the addition of poly A tail. Once the poly A polymerase have acted, then the RNA is now having a poly A tail, functional poly A tail. So with this, I end this topic of transcription termination, and we will resume in another class. Thank you.